Welcome to another screencast. Well, this time I'll be discussing a little bit about the ventricles and the cerebrospinal fluid of the brain here. So by the end of this, you should be able to identify the location and structure of the four ventricles of the brain, as well as be able to discuss here a little bit about the production, composition, and circulation of the cerebrospinal fluid, which we'll just be referring to as the CSF. Okay, so within the brain, there are these ventricles, or these little receptacles, or little hollowed out cavities, if you will, that will contain the cerebrospinal fluid. So if we look at a lateral view here of this, within the two cerebral hemispheres, you have these little pockets of space that are going to be filled with cerebrospinal fluid. So the two that are sort of within the hemispheres are the lateral ventricles. And so there's a right lateral ventricle and a left lateral ventricle. So these are two of the four. The third is commonly or easily referred to as the third ventricle, and we see that structure here. If we look at an anterior view, that third ventricle is going to be located right here, and the tissue that surrounds that on either side is going to be the thalamus. All right, so the thalamus is on either side of that. So when we make a mid-sagittal section through the brain, you're sort of cutting right through that third ventricle there. So if you're looking at a medial view of a mid-sagittal section through the brain, the third ventricle, you would have cut right through it. So that'll be the space where you'd put the probe before you were to actually touch the thalamus there. Right. So these two lateral ventricles are not connected directly to one another, but they are connected via this interventricular foramen. And again, foramen is a hole. And so this interventricular foramen right here and right here is actually more part of the third ventricle, but there isn't any sort of like gross connection between here, the right lateral ventricle and the left lateral ventricle there. So they are continuous with one another, but just at that interventricular foramen. The fourth ventricle is then located down here, just anterior to the cerebellum. And so we see this fourth ventricle area here, and we see it grossly here. And that third and fourth ventricles, those third and fourth ventricles are connected by the cerebral aqueduct. Aqueduct, of course, a passageway for fluid or for water. Right? So that cerebral aqueduct connects the third and fourth ventricles. Right? And so this gets us, the third one, and here's the fourth of the ventricles. Okay, so let's take another look at now here this. So again, when we make a mid-sagittal section through the sheep brain, this here is the thalamus area, and this area, I'm not touching the thalamus here. Right? So now I'm touching the thalamus. Now I'm not touching it. Right? Touching, not touching. Not touching the thalamus, that area would be the third ventricle there. To see the lateral ventricles, you would put the probe up underneath this corpus callosum there, and we may have to make just a sagittal section, not a midline section through the brain, in order to see those lateral ventricles, because that's all going to be up inside of, more lateral to that corpus callosum and in the cerebral hemispheres there. We can then identify here the cerebral aqueduct down into where the fourth ventricle is going to be, right in front of that cerebellum there, or anterior to the cerebellum. So if we look at what's in these, what's in them is a fluid called cerebrospinal fluid that is going to be constantly circulating, surrounding, and bathing the exposed surfaces of the central nervous system. The cerebrospinal fluid is produced by specialized knots of capillaries that are called choroid plexuses. A choroid plexus. Right? And so there are several of these choroid plexuses around. So the extension of the choroid plexus into the lateral ventricle. So we're going to have this knot of capillaries here, and on the sheep brains is going to look really dark and black. So one is this one is sort of coming out at you in the diagram. This one is going back into at the base of the the lateral ventricles there. So this one that I identified here is the choroid plexus of the third ventricle. And then we have another choroid plexus down here in the fourth ventricle, which is where the cerebrospinal fluid sort of is being produced. So that's why you can follow these red arrows there. So the cerebrospinal fluid is going to then constantly be circulating because of ciliated ependymal cells. These ciliated ependymal cells are modified epithelial cells, right, that are lining these cavities, right, and the cilia on them keeps that cerebrospinal fluid circulating. So if you were to zoom in right here, say, you would see ependymal cells that are ciliated that are going to keep that fluid moving. There are a couple places here where so this fourth ventricle then becomes continuous with that central canal of the spinal cord, and this break is just showing you that it's a longer structure here. So that central canal has the cerebrospinal fluid in it, and that's circulating. And then we have these little ports, right, this median aperture here is where that cerebrospinal fluid then can be circulated there around the outside surface of that nervous tissue as well. And so you're following the red arrows here. It circulates around the cerebellum, and what's keeping that fluid from leaking out, of course, is the Dura Mater. So here's the Dura Mater, right? And so that cerebrospinal fluid is in that subdural space, right, between the arachnoid mater and the Dura Mater. So all of this darker brown stuff right here is the subdural space. So if they do a lumbar puncture or a spinal tap, this is where they are entering with the needle. They're going to puncture that Dura Mater and then remove some of that cerebrospinal fluid. So this would be the needle here. They would remove some of that cerebrospinal fluid down there to check its contents, to check its composition, make sure there aren't any bacteria, viruses, fungal spores, or anything that happen to get in there, that happen to cross this blood-brain barrier here at the choroid plexus, or those capillaries. One final structure we want to point out here is that the cerebrospinal fluid then is returned to the bloodstream at these arachnoid granulations, right here. So if we go into the next image here, we're going to zoom in just on those arachnoid granulations. So we're looking here. And so what we see first is the Dura Mater. Right? Here is this is the meningeal layer. Here's the periosteal layer. And so we have the superior sagittal sinus, and this sinus is a modified vein, and sometimes described as being venous. Right? And so the cerebrospinal fluid is going to be moved and across into these little granulations, and the cerebrospinal fluid is filtered across these granulations, which are little extensions of the arachnoid mater, up into the superior sagittal sinus here. And as that fluid enters the superior sagittal sinus, it will be then circulated back here into a vein and then returned to the bloodstream this way. And so this superior sagittal sinus, again, this sinus is an opening, is a space that is going to contain cerebrospinal fluid there. But then that cerebrospinal fluid moves moves anterior, but then primarily is going to move posteriorly and then re-enter the bloodstream there as part of the plasma and get filtered back around. So this CSF constantly moving, right? You only have a total of about 150 mLs of cerebrospinal fluid, and it's constantly being circulated. So this means that your CSF turns over about 8 times per day. So you replenish that roughly about 8 times per day. Okay. So hopefully here you're able to describe and identify the location and structure of the four ventricles, as well as the little connections between them, the interventricular foramen, as well as the cerebral aqueduct and the central canal of the spinal cord, and then be able to discuss here a little bit about the production, composition, and circulation of that cerebrospinal fluid. Okay, thanks for listening. Bring your questions to class.